All right, class. Now we're going to discuss, up to now, in this entire chapter 3, the cell, we've discussed the normal activities of the cell, which would have been under physiology. However, you're going into healthcare, so we at least need to glance at some of the things that go wrong. Let's pick the most ominous situation that can occur, and that's what we call tumors. Tumors basically is cell division gone wrong. Cell division with no controls, unlimited, that has gone wrong. Causes of tumors can be genetic. All tumors actually, let me make a better statement. All tumors are genetic, but that does not mean hereditary. So all tumors have to do with some mutation in a gene, but that does not mean, genetic does not mean hereditary. The causes for these genetic problems are numerous. A growth factor oversecretes. Growth factor being where one cell tells another cell to activate. A cell itself becomes autonomous. A proto-oncogene becomes mutated to an oncogene, or tumor suppressor gene fails to work. The genes we said they control for cell division in the actual cell itself or in a neighboring cell or a cell even further away that tries to tell this cell to divide are called proto-oncogenes. Any gene making proteins associated with the function of cell growth are called proto-oncogenes. A tumor suppressor gene is kind of like an anti-oncogene in which it rides herd, old Texas term, over the proto-oncogene so they don't go out of control and have abnormal cell growth. So a mutation of the proto-oncogene could potentially make it an oncogene. Now, mutations are spontaneous effects. So the mutation in the proto-oncogene could make it less effective, in which case that you'd have a decrease in cell multiplication. Now let's look at we have the three terms are normalplasia would be a normal amount of cells being made homeostatically. Hyperplasia, too many being made. Hypoplasia, too few. So a mutation in a proto-oncogene making it less effective would give you hypoplasia. But if it made it too strong, it could give you hyperplasia, abnormal, which would then take you to becoming what we call an oncogene. A tumor suppressor gene, like an anti-oncogene, if it becomes less effective, that's when the problem occurs. It's a mutation. You can look at that sometimes as something that, so proto-oncogenes can be converted to oncogenes. And we won't get into this. You're not as familiar with DNA, but we have different types of mutations and so forth. Okay. Here's some proto-oncogenes include the RAS, the Ralph sarcoma, and so forth like that. Again, this is a little bit beyond you, but I wanted to, this PowerPoint again is for not just your class, but classes also more advanced. A little bit ahead of you there, showing different growth factors that would be coming from one cell telling another cell to turn on. Okay. Tumor suppressor genes, they ride herd over the other. Repression of genes are essential for continuing of the cell cycle. And so there are various reasons for these. If the damage can be repaired, the cell cycle can continue. This kind of holds back the cell cycle if there's DNA damage. So there are various reasons for great tumor suppressor genes. To help prevent uncontrolled cell growth, tumor repair damaged DNA, control cell adhesion, various things that they do. All of these in an attempt to not let things get out of control. Tumor suppressor genes. Examples of the most notable one is the P53 tumor suppressor. And that has a lot to do with various cancers, colon cancers, breast, and so forth like that there. That's a tumor suppressor gene. The first tumor was the retinoblastoma. Retinoblastoma is in the eye, on the retina. I won't get into the two-hit hypotheses. Here's tumor formation. Here would be the cell cycle overstimulated. What would that be? Proto-oncogene becoming oncogene. Cell not inhibited. What would that be? Tumor suppressor gene not working. Increased cell division, abnormal, hyperplasia. A tumor, to say another term, is an abnormal hyperplastic process. A tumor is an abnormal hyperplastic process, remembering that hyperplasia is an increase in the number of cells. Now, you could have normal hyperplasia, but a tumor is abnormal hyperplasia. Here's kind of a understanding progression of colon cancer. Normal colon epithelial cells, we haven't done, those are cells on the surface. Loss of tumor suppressor gene. Small benign polyp is a growth. Now that's benign. Activation of this here, this oncogene, loss of this tumor suppressor, and there you are. What exactly is a tumor? Well, Stedman, which writes dictionaries in healthcare and so forth like that, science, is any swelling or tumefaction, also called a neoplasm. Now that means it could be a solid swelling or cystic. Generally though, when we consider a neoplasm, new growth, it's generally a solid most of the time. But it could be a swelling according to Stedman, but it's generally a solid. A neoplasm is a tumor, an abnormal tissue that grows by cellular proliferation, that means multiplication, more rapidly than normal and continues to grow after the stimuli that inherited the new cell growth ceases. It goes autonomous. Okay. Here's a comparison of benign and malignant. Benign tumors generally are slow. This is not always the case now. Expansion. You can read that yourself. A benign tumor, though, when if you have a tumor and you say, you go to the doctor and they say, well, it's benign after we biopsy, it still can have problems. I wanted to point that out. Major problems with benign are space occupied. They will crowd out good functioning tissue. Pain, depending on where it is, it can give pain. It can give pressure. It can give bleeding if it cuts into the wrong area, and inflammation. So there are problems that we oftentimes have to take benign tumors out. So I don't want to make it seem like, but the key is that it can't do this I'm going to talk about, called metastasis in a minute, which means to move from one site to another site and start the same deleterious process there. So that's benign. So cancer comes from the word crab. Hippocrates, one of the fathers of medicine, it looked like a crab grabbing in. When we look at the cancers, we group them into carcinomas, sarcomas, and lymphomas. A carcinoma, which when we get to chapter 4, we'll talk about different types of tissue. A carcinoma is a tumor of epithelial tissue. We got 4 different tissue types we'll talk about in A&P in the 4th chapter. One is epithelial, connective, nerve, and muscle. Those are the 4. Epithelial, connective, nerve, and muscle. A carcinoma is one of epithelial tissue. A sarcoma is one of connective tissue, and lymphomas are blood and lymphatic origin. We mentioned metastasis. Metastasis is a situation where the tumor can spread to distant sites or even adjacent sites. Okay? They can invade surrounding tissues and vessels, so that's neighboring site. Decrease in cells break in cancer. Cells break free, and they can travel to distant sites. So there's a break free of cell to cell, increased cell mobility, and that distant site can go via blood vessels or lymphatics. Some cancers spread better through blood vessels and some through lymphatics. Okay. So where the cancer started just to show you a little illustration, is the primary site. And where it moves to is called the secondary or metastatic site. Secondary. So it metastasized to this area. Okay. Characteristics shared by cancer. Cancers have a distinct appearance. They look bizarre under the microscope. They don't get back to differentiation. Why? Because they spend little time, go back and study in G1. G1 was when they did differentiation after splitting into two cells. They go straight into S fairly quickly. They are immortal in culture. That means you can grow them forever almost in a culture. The famous HeLa cells, Helen Lane, Henrietta Lacks. We've been doing there's been a lot of book reading in that over the last 2 or 3 years at the college here to show about that. But the HeLa cells were from a lady that had female cancer, uterine cancer, and they just kept growing them. They lose contact inhibition. Generally, when cells are on a culture plate, once they touch one another, they stop. So they spread out over the whole plate. But with cancer, they just keep multiplying. So therefore, if you've already spread out the whole plate, where's the next one going to go on top of those? They can secrete a transforming growth factor, which can cause other cells to start doing the misbehaving they did. They lack normal cell cycle controls. Go back to your cell cycle control. At the end of G1 was the restriction point. They lose that. Cell surface alterations. So, and then tumor markers. I may get a chance to speak on that. Tumor markers. They may secrete tumor markers. Oh, well, I am speaking. Okay. Tumor markers are, think about this, where there's smoke, there's fire. Where there's smoke, there's fire. So if you see the smoke, you know the fire is someplace, even if you don't see it. What a tumor marker basically is a substance that we can draw in the blood. And certain tumors will secrete small amounts of this in the circulation. Now, some of these tumor markers are there normally. But so in that case, the increase in amount would say, there's a tumor going on. So like for prostate cancer, we draw this PSA in males. Even when the prostate cancer is small, you may have an elevation in this. This is another one, CEA, which can sometimes increase in colon cancer. Now, that doesn't always mean you have colon cancer. It's indicative. Even with this, if it's up, does not always mean you have that, but at least it makes the physician, the clinician, check, now do very detailed studies. Alpha-fetoprotein or other. So I'm just showing you a few. So when you have these tumor markers, now risk markers, on the other hand, are things you draw in the blood, and it says because you have that, you have an increased predisposition to certain cancers. This is a major one that all you females need, the BRCA, the breast cancer one. The breast cancer one is a marker. What it is it's a mutation in tumor suppressors, which means you make this, and this is a mutant tumor suppressor, BRCA. So if you have this BRCA, now they are actually BRCA1 and 2 genes. If you have that, you can see if they find that gene, an increased chance of breast and ovarian cancer. And so both of these have a slightly increased risk. So this is what we call the BRAC. So we talked about tumor markers and risk markers, measuring for risk markers. Okay, we will close here. This will be tumor video 1. And now we're going to tumor video 2, and that will be causes of cancer and subsequent talk.